And Ryan, I appreciate the opportunity to talk today, and I appreciate everyone joining in. I hope to share a few things with you. And I've changed the topic a little bit to make this more usable, I think, over the long term for everyone in their operations. And so what I'm going to do is I'm going to point you to different references and materials that you can download as well throughout the talk. And I'm going to try to put the websites as much as I can on the top or the bottom of the screen and stay consistent so you can get more information that way. And if I understand right, this is recorded, so you'll be able to come back to it as well. So with soybeans, really I tend to think of them as having diseases in the root, stems, and leaves. And soybeans are quite a lot different than many of the other crops that we work with. The root problems are far and away the most important issues we have with soybean diseases. We do see some diseases in the stems, and Greg, of course, just talked about white mold. We do see other ones show up over time, but honestly, I'm not too concerned about many or any of them in the West. And it's unusual that you got hit with white mold so bad this year, but we do need to talk about that one. And then with the leaves, we pretty much all but completely ignore lesions on the leaves. We just don't have something that even resembles a rust. I mean, there is a soybean rust, but it's in the Gulf states only. We don't have things like septoria or tan spot on wheat. We just don't have them. What we do see on soybean leaves occasionally is a little bacteria and a little bit of septoria. And in our area, we don't even worry about those for yield issues. So we'll start a little bit with diagnosis before we get into some of the root rots and white mold. A diagnosis is really important, especially if you're newer to soybeans. So you'll see things that you might not have seen before. And so to try to get something into your hands that you could look at in the field that was field quality and try to identify what was going on, the North Dakota Soybean Council and the Minnesota Soybean Promotion Board put together, or gave us funding to put together, a diagnostic series that was specific to North Dakota and northern Minnesota. And the way we organized this is we grouped them into root diseases, stem diseases, and leaf diseases. And the concept here is that you can take this into the field and you can quickly flip through them and hopefully identify something. And it's available up here at this website. And there are print copies available at the NDSU print and copy service, and you can access them here. And the ones that are printed out are not paper, they're really high quality. So as long as NDSU is still operating and people are in the building, I think you could request some, or from your county office, your county extension agent. So the way these are designed is the front side is going to all be photographs, right? So you can see white mold here. There's several pictures of white mold, things that are diagnostic to white mold. And then the back side is going to be things that are maybe critical for diagnosis that might not be intuitive. So symptoms is obvious, but factors favoring development maybe is not. And then important facts below, you have ideas where maybe you can manage it a little bit, maybe you can put something into a structure to diagnose it, maybe you need to look for something specific. It's really all based on diagnosis. So here, and these were animated, but the idea is you flip through a bunch of these different cards, just the front side, you can look at the symptoms and see if anything rings a bell. And then flip them over, and you can compare those symptoms, those factors favoring development, and some of the important facts. And just as an example, I'll highlight this in the middle. This factors favoring development is usually very important, and I'm just comparing white mold over here to charcoal rot over here. And you can see that charcoal rot likes hot temperatures, drought stress, and maybe where SCN is present. But white mold, it's going to be wet, it's going to be cool, and you're going to usually have disease history in the field. So even though they might look alike from a certain distance, this will help you figure out what you've got going. All right, so we'll switch into root rots here. So with root rots, we have a lot of different pathogens. This is kind of normal for plant diseases, although I would say that soybeans tend to have more root rot pathogens than most. And it's an interesting scenario. You don't see a lot of root rot damage when you first get into soybeans, but over time that becomes a big issue. And so you can see these Pythium, Phytophthora, Fusarium, Rhizoctonia, you see these pathogens floating around, especially in eastern North Dakota, and eventually in western North Dakota. Of course, you'll see more damage for things like Phytophthora, which you probably don't have much, if any, right now. So soil moisture conditions really range from flooded to dry though, and soil temperatures that are favorable for these range from cool to warm. So the message here really is there's a root rot pathogen for every environment. Certainly you see more root rots when you're wet and generally when you're cool, but you can have problems when you get hot too. So they're broken into two biological groups. So Pythium and Phytophthora are really not true fungi. They're actually more related to algae. And they look a little different. They look a little bit like slimy root rots or slimy roots or maybe a little bit of a blackened lower stem. But the important part here is that they need wet soil. And they need wet soil because they have swimming spores. And so if you ever have seen downy mildew in sunflowers, or maybe a Phantomyces in field peas, it's the same general group. So each of these, if you get water after planting, and especially in Pythium's case, if it's cool, if you get water after planting, you often will see these show up. And the same is true, of course, with downy mildew. And if you've grown sunflowers, you know if you get rain after planting, you're going to see that. Fusarium and Rhizoctonia are in an entirely different group. They're the true fungi, and the lesions look a little bit different. Sometimes they're reddish. They kind of look like a dry rot. And these would be fungi, same as like bread mold, or same as many of the other things that we see. And it's notable here that Fusarium, the genus Fusarium, is really huge. So we talk about Fusarium head blight in wheat. That's generally 1 or 2 or maybe 3 species. We don't generally worry about those species in soybean root rots. There's just hundreds and hundreds of species that we see in the field. So I had a video clip, and I'm going to try to play this if it will play on your screen. So what you're looking at is the swimming spores that I mentioned, and this is Phytophthora root rot in soybeans. But you can use your imagination. This could be the downy mildew pathogen or Phantomyces in peas. So what's happening here is you have this reproductive structure, and those spores have tails. And when they break through that structure, they will swim away, and they are chemotaxic. So they will follow root exudates, and they'll follow chemical trails. It's quite an amazing thing. But I put this up here to try to help explain why we have root rots, really, when we have a lot of water in a field after planting. So this is a soybean root, and this is also done in the lab. And if you look at the center of the screen, you've got that same reproductive structure releasing spores. And all this other foresty looking white stuff, that's all the Phytophthora pathogen. So the Fusarium and Rhizoctonia, they don't have spores like this. This is really a waterborne, related to algae pathogen. All right, so if we're looking at managing root rots, and we're thinking about root rots, the very most important and first thing I always would say is rotation is key. So Pythium and Fusarium tend to be fairly broad. Okay, so if you have Fusarium damage or Pythium damage in a different crop, there's a little bit more likelihood you're going to get it in soybeans too. And I'm using the word broad in general terms, right? So Fusarium, one species isn't going to affect everything, but it's because there's so many species, and Pythium is the same way. So you can get Pythium in pretty much every crop. It might not be the same species, it might not be the same severity, but they will show up in soybeans, and they'll show up in dry beans and sunflowers and canola and other things as well. Rhizoctonia is semi-specific to the crop. And so this is important if you, which I don't think many of you do, but if you had dry edible beans or maybe sugar beets in a rotation, which would be much more rare for the west than the east of the state, you would be more concerned about Rhizoctonia. So Rhizoc is semi-specific. And the reason is that Rhizoc has some genetic differences where a Rhizoc in soybeans won't actually hit wheat. A different Rhizoc might, but that same one is mutually exclusive. And the last one to consider is Phytophthora is specific to the crop. So Phytophthora really only affects soybeans. There are other Phytophtheras floating around, but Phytophthora sojae is the soybean pathogen, and it doesn't go to anything else. So what I'm putting this up here for is to get you to think about the similarity of crops when you're planting in rotation, right? So the closer related a crop is, so for example, a legume broadleaf crop is probably more likely to share some of the same Pythium or Fusarium pathogens that soybeans will. Whereas if you go to a crop that's quite a lot different, so barley, for example, they will share almost none. So the further away, the less similar the crops, the better the crop rotation is for disease. And that is true with almost every disease. There are rare exceptions. The next thing is how to manage these root rots. And Phytophthora, if you have Phytophthora, there are specific resistance genes for Phytophthora that are quite effective out west, where we're losing them in the east. But if you see Phytophthora, if you scout it, you figure it out, you know it's Phytophthora, the next time you go back into that field, pick a good solid gene and you'll be much better off. There's also field tolerance and resistance, so maybe defensive beans or ones that have field tolerance to root rots, they're quite good as well, and that's something you'd want to consider if you start to see root rot. And then the last thing I would mention is that we have some seed treatments. And to be frank here, seed treatments are a really good investment for soybeans. The fungicide part, they're really fairly inexpensive. They protect a seed, which is a really big investment. And they do a really good job in general, the fungicide seed treatments. The insecticide ones and the nematicide ones get increasingly more expensive and increasingly a little bit more questionable if you're getting your money back. But generally the fungicide ones are. Unless you're just getting into soybeans the first year or maybe the second time around, you might want to consider a seed treatment. If you're just new to soybeans, you probably just really aren't going to see a lot of root rot issues. It takes time for them to build up. But with seed treatments, you want something that's broad spectrum. And what I'm talking about here is you have these two different groups. You've got the Fusarium and the Rhizoc, and you've got the Pythium and Phytophthora, and one of those chemicals is not going to manage both, right? You just generally, because one's related to algae and one's fungi, they work different, the chemicals work different. I'll show you that. So you want a blend, and we'll talk about that a little bit more. And then in general, seed treatments are only effective for a few weeks, but they're an important few weeks. Germination, stand establishment are pretty critical. All right, so you'll have to bear with me here. This was all animated, so I'm going to try to take you through some of these websites. So the first one is this cropprotectionnetwork.org. And what the Crop Protection Network is a group of mostly extension plant pathologists from land-grant universities in the north, central, and southern states. And so what we've done is we put together a bunch of data, we put together a bunch of resources, and created this website where it's like a clearinghouse for a lot of multi-state work. So certainly NDSU has their own extension publications and webinars and things like that. This is designed for broader acre crops and bigger problems. And so I would encourage you to take a look at some of this. And again, that website is going to stay up here, the Crop Protection Network, I think, for a little bit, unless I animate over it. So what I'm going to do is I'm going to take you through publications, this first yellow arrow. And publications, I'll show some down here. You get there by clicking on the resources. So go to Crop Protection Network, click resources, and you'll see publications drop down or publications here. And I'm going to show you a couple of publications. So there's 3 yellow arrows here. The top yellow arrow is a publication on factors to consider before using a seed treatment. Not everyone needs a fungicide seed treatment, especially if you're just getting in, but over time they become very, very effective. So that's maybe a good publication to take a look at. The next one is Fungicide Efficacy for Control of Soybean Foliar Diseases. And so how this works is a publication, actually the next 2 arrows really, we put our data together and then we try to determine how effective these different chemicals are against certain pathogens. And I have an example here. And then this last one, Fungicide Efficacy for Control of Soybean Seedling Diseases, is probably the most important. And this is the one I'm going to show you. All right, so this publication, and you can access that at the Crop Protection Network, this is what it looks like. And so we have this regional group, actually it's bigger than a region, and we put our data together and we evaluate some of these different chemistries for effectiveness. Okay, so this is the table, part of the table that's on the back. And the way you would read this is you've got active ingredients here, so these are all the fungicides. And then across the top, you've got Pythium, Phytophthora, Rhizoc, Fusarium, sudden death syndrome, and Phomopsis. The last 2, these 2, we don't need to worry really about in the West at all. We just confirmed this one in Richland County just last year. This one is around, but I don't really see any yield losses coming from it. So, but these 4 are pretty important. Okay, so remember I said that some of these chemicals work really well on those algae-like pathogens, and some work really well on the fungal pathogens. The color of the arrow is examples of how that works in the real world. So here you have a yellow arrow and a yellow arrow at ethoboxam and mefenoxam. And if you look at Pythium and Phytophthora, we've rated ethoboxam and mefenoxam E for Pythium and Phytophthora control, and E as an excellent. They work really, really well on those two algal-like pathogens. However, you go across to Rhizoctonium and Fusarium, we don't recommend them at all because they don't work at all. And the reason is that the mechanism, the mode of actions in ethoboxam and mefenoxam are targeting that specific oomycete biological structure, which don't exist in Rhizoc and Fusarium. Okay. The flip side is some of these other ones, azoxystrobin, fludeoxanil, fluxiperoxide, and conazole. They don't work very well on these Pythium and Phytophoras, but they work pretty good on Rhizoc and Fusarium, right? So the idea here is that if you're looking at a fungicide seed treatment and you're looking at it for soybeans, you probably have, or at least over time, all these things. And what you want is not just one compound, you want a blend. You want something like Apron Max, not just Apron, not just Maxim, you want Apron Max, right, as an example. So you need a blend to manage each of these together. Otherwise, you might be missing half of them. Okay, to help you take a look at this and figure out what's actually on your seed, I'm pointing you to another extension publication, and this comes out of the University of Wisconsin-Madison. And I know this is jumbo, that was going to be animated. I apologize for that. So if you go to this website and take a look at this publication, there's a legend on the left-hand side where the Wisconsin extension people have categorized these just by color: fungicide, insecticide, nematicide, plant growth regulator. Okay, so you can quickly look over here across the top. You've got active ingredient, the trade name, and the crop. So Wisconsin doesn't really care about some of the crops we grow like sunflowers, but they do have small grains and soybeans and corn, so those are fairly important. Okay, so if you're going to read this, so you've got azoxystrobin, right? That's an active ingredient. What is it labeled as? Dynasty. And then you can probably ignore this because you have other resources to figure out if it's labeled on soybeans or flax or whatever. All right, and I should say, this is not soybean specific. These names often go across all the crops we grow in North Dakota. So this is pretty simple, but you go down here and you ask yourself, well, what the heck is in Maxim Quattro? Well, you can see 4 different fungicides. Or maybe Cruiser Max cereals, what is that? Well, you can see it's mefenoxam, difenoconazole, and thiamethoxam. Okay, this helps you decode it. So mefenoxam works really good on some of those Pythium Phytophthora-like species. Difenoconazole works pretty good on some of the others that we talked about, and thiamethoxam's an insecticide. So this publication, I'll be honest, I keep it by my phone because I can't keep these things straight. So this is something that might be helpful when you're looking at products. All right, the next one I wanted to move to was white mold. And so white mold is something you got hit with fairly hard in the West this year. It's kind of unusual to be hit as bad as you were. And it's unfortunate, and I hope it doesn't happen again. But we'll go over a few things that you can do about it. So the first is that it's a fungal pathogen. And it's called Sclerotinia sclerotiorum. So if you ever hear Sclerotinia white mold or sclerotia, being the little black structures, it's because of the pathogen, how it's named. But you get yield loss, and you need to manage it with an integrated strategy. There's not a silver bullet for white mold. There's really not a silver bullet for white mold on any of the crops we grow. And the other column here, I've got sunflower and dry beans and canola and soybean, pulse crop, and broadleaf wheat. So white mold is kind of unique in that it hits about 500 different plant species. In North Dakota, it will hit every single broadleaf crop we grow. The only one that might be considered resistant is flax. And the most susceptible is probably sunflower, followed closely by dry edible beans. And soybean is in there, it's in the mix, but it's not like a sunflower. So same pathogen, no races, exactly the same thing is going to hit all these crops. So if you had white mold last year and you're in say a sunflower crop and you're thinking about putting soybeans on, or you fill in the blank, right? If you had white mold in that field and you're thinking about soybeans, white mold is probably a concern because of this particular pathogen's host range. Okay, so when do you get white mold? So I mentioned that it's on almost every broadleaf crop, so I've got some photos to use as an example of these different crops to try to show you the story here. And this again was animated, so it might be a little choppy. So the first thing you need is you need wet soil. And generally 1 to 2 inches of rain before bloom is probably what you need as a minimum. If you have more, you're more likely to get more white mold, and it's all related to the biology. So what we're looking at here is we've got a bean that I grabbed, and it's got sclerotia tucked inside this bean. And these, of course, occur in the stem or the head of a sunflower or wherever. So if you have those on the surface or within about 1 inch of the surface in the field, and you get that 1 to 2 inches of rain, they will germinate. And when they germinate, they produce apothecia. And apothecia are this mushroomy looking structure, and they kick out ascospores. And so at this point, if you had the water, you got these things growing, now you have ascospores floating around the sky. And that is a little bit of the weak point. The ascospores don't infect healthy green tissue. What they do infect is florets. And so here you can see a sunflower leaf that I found in the field, and there's a floret right in the center. The ascospores hit that, and then from that point, then it grows into the healthy green tissue. So flowering is critical for white mold, right? We don't even think about plants being susceptible, with the exception of sunflower maybe, before the flowering time, okay? All right, for this to happen, you need it cool and wet. So you need 60s, 70s degrees, and you need a long, humid canopy, frequent rains, heavy dews, things like that. And a thick canopy also makes a difference. And I heard Greg talk a little bit about other things related to white mold, and he's absolutely right. Thick canopies make a difference, although I would not recommend changing something like row spacing to manage white mold. We'll talk about that in a little bit. All right, so once you go here, you can pretty quickly go from wherever the infection started on the floret right into the stem or the branch, and it'll rub out that plant pretty quick, and then you'll have these black structures, these sclerotia again. Okay. All right, so managing it. So cultural practices, it's a mixed bag. Tillage is a mixed bag, right? So the reason is if you leave sclerotia on the surface, they're more likely to germinate that following year. You get big flush of spores, and then a lot of them are gone, which is good. If you bury them down, they'll survive, and then when you bring them back up, you're going to have those little mushrooms form and spores. So it's a mixed bag. Row spacing, yeah, Greg talked about that a little bit. Michael had some data that suggested, yeah, in wider rows, you get less white mold under high pressure. And I don't see really any spot in this state where it's a good idea to risk losing yield because you're widening and widening your rows just to deal with white mold. So I would not recommend that. Use the rows that work for your operation, chase the yield, and we'll deal with white mold later. That's not the best management tool. Rotation is really important. Cereals and corn are the best rotation crops. Remember, all broadleaf crops are susceptible. Sunflowers have a special place in this because they can get infected through the roots, so you have to be particularly careful with sunflowers, but rotation is really important. And then resistance. So there is some partial resistance. I'll mention that in a little bit. And then fungicides, of course. And fungicides, you ask yourself, how favorable is it for infection? Do we get an economic return? There's a lot of products out there that can work, and timing is important. And I'll show you some resources here that'll help you. Okay, so the first is that we're going to go to the Carrington Research Extension Center plant pathology page. So Dr. Michael Bunch is the plant pathologist at Carrington. He is an excellent white mold researcher. And so you can get to this by clicking into that website or chasing that website, or just Google the Carrington REC. And then this arrow down here shows you the plant pathology tab. Just click on that and this is what you get. So Michael has updated a lot of different data since, well, I guess it says St. Patrick's Day, March 17th. So he has information on multiple crops. There's sunflowers, there's optimizing, there's soybeans, there's dry beans. There's all kinds of white mold information here. It's like a white mold clearinghouse. It's great. So the information that I'm grabbing is coming from this site. And Michael is a very approachable individual too if you need to talk to him. All right, so I mentioned that there's some genetic resistance, and it's partial resistance. And so one of the things that Michael's done at Carrington is he's taken a look at these different varieties. So you have a variety column here, And he's evaluated them against white mold severity here and yield here. And you can see the white mold severity, the center column, goes from basically having virtually none to having quite a lot, over 50% of the plants infected. That's pretty bad. And you can see that it correlates really well to yield. And that's a doubling effect of yield for white mold. So my recommendation is, if you're thinking about varieties, you haven't made the decision, you had white mold on that field, see if you can find a little data on genetic resistance. It's not perfect. The company information is not perfect. It's very difficult to screen for white mold. You need a natural epidemic. Michael's got some good data here, but you can see he's probably got 40 or 50 varieties, and there's quite a lot more you can choose from. The best information you can get on varieties is going to be helpful to you. And I would encourage you to chase that down a little bit if you haven't made a decision yet. The other thing that he's got a lot of is he's got some fungicide efficacy, and this is just an example, right? So there's a lot of data on there. I'm pulling this example from 2013. This is on soybeans. This is under very high disease pressure. I want to emphasize that. This is very high disease pressure. It's probably unusual in most of the state, but what it does is it gives us a really good example of how these products work under duress, right? So here in this column, you've got these different fungicides, and notably there's two applications here. There's different application timings that people are playing around with. So they're here. There's a couple different rates involved. You can take a look at this or other data on the site, but you got the treatments here. You've got sclerotinia incidence here, and you've got soybean yield on the far right. And you can see that some of these fungicides do work fairly well. Now there's a lot of pressure in here, right? This helps separate out efficacy. That is true. I don't know that I would say that you'll see this on your farms most of the time. And in rare occasions, yes. And in those occasions, yes, fungicides will make sense. The last thing that I'm going to share with you relates to decision-making timing. Okay, so this is a complicated slide, but it's very good information. And I'm just going to kind of summarize this. So in the past, we really looked at growth stage as the timing, decision-making timing, right? Growth stage was really important. And the reason we care about growth stage is that when those plants start to bloom, they have those flowers, and then they become susceptible to white mold because that's what the spores land on. So you don't want to go too early because you'll miss it and won't protect them. You don't want to go too late because all the early infections have already occurred. Well, what Michael's found, and others have thought this, but he was really the one that did the experiments to document it, is that it's not just bloom, it also relates to the microclimate and specifically canopy closure. Okay, so he's kind of got a three-part recommendation going on. So it's all based on canopy closure. So under favorable disease environments, when the canopy was closed at mid to late R1, you probably want to hit it with a fungicide right then, because the microclimate at R1 is when the blooms pop, and if the microclimate is in line, that's probably the right timing most of the time. If the canopy was starting to close at early R2, and you have a favorable environment, that's when you want to hit it. So it's more about canopy closure than it is R1 or R2. It's really related to the canopy closure. And then at full R2, even if the canopy wasn't closed, that's the time to hit it. And the reason is if you wait till R3, you probably have too many infections already, even without the canopy closure, that you're going to miss the window. Okay, so the way I think about this is that at R1, you look at the canopy, and when it gets close to being closed or closed, you hit it. Unless it doesn't even close until R3, and then you missed it. You hit it at R2. But again, this is only under high disease pressure, only under favorable disease environments. Another tool that you can take a look at to help you make a decision, and this is the sclerotinia risk map. So this was funded by the Northern Canola Growers, and it was developed, gosh, I want to say almost a decade ago now. And how this works is it takes into account end-on data from across North Dakota and northern Minnesota. It looks at the biology of sclerotinia. So are there spores in the air? Do we expect favorable environments? Things like that. And when you are hot for sclerotinia or at risk, it's red. And when you're low, it's green. And anything between that is shades going from green to red. And so here I just pulled out the image from the 4th of July. It's a great day. except it seems like it's always raining. And of course, in this particular case, it was very favorable for white mold across most of the state this last year. And so this is available at this website. It's active, it's not active now, well, I think it is, but it doesn't make sense. So it's active all summer long. And you wanna look at this probably right before you get into bloom and then through bloom. It doesn't really matter if your plants aren't blooming what this actually says. So really it's a little bit before bloom and then bloom. Okay, Ryan, I'm not sure how much time I have. I thought I would touch on SCN if we have time, and if we don't, that's not a problem either.
Yeah, if you just want to touch on it briefly. We started you a little late, but we're about where we should be wrapping things up here in a little bit. So.
All right. The last thing SCN is important. It's the last thing I'm going to talk about, but I want to emphasize this is a problem really for the future. But if there's one thing I can encourage you to do is start to think about soil sampling for SCN. So if you've been growing soybeans for a while, you really want to think about this. I'm going to show you why relatively quickly. So there is a website where you can access a ton of information called the SCNcoalition.com. The SCN Coalition is a group of a bunch of different land-grant universities and a bunch of private companies, so BASF, Bayer, Syngenta, things like that. We're trying to unify our messaging because SCN is causing $1 to $2 billion of yield loss a year in the US. It's racking our brain pretty hard in the East. It's not in the West yet that we know of, but that's why I'm asking you to sample. So just real quickly, it's a parasitic worm. That's the worm. The root, it looks like this. So you've got little cysts. These are little female worms. You compare that to a nodule. They're really quite small. And then it's an invasive pathogen. So this is not from North America, and it's spread on anything that moves soil. So in the 50s, it showed up and hit the Boot Hill, Missouri. By the 70s, it started to spread through the Mississippi River Valley. By the 90s, you had hot spots all over. By 2000, it was just knocking at North Dakota's door. And then this map from a couple years ago shows the states where we have confirmed SCN. So to sample, there is a sampling program that the county office is engaged in. And how this works is the North Dakota Soybean Council funds a whole bunch of lab fees and prepaid sample bags. And so what you'd want to do is go to the county extension office or REC, get some sample bags, and just sample for it. And there's instructions that can be sent with you. And the data is reported at 100 cc. So let's look at North Dakota here quick. So this is a map that we've been populating based on these samples. And to be clear, this is growers and consultants who've been sending them in. I don't see any names. I don't see anything except the data point to map. All right. These are all the negatives. So there's about 3,000 negatives, right? But this is good to show where things have been sampled. These gray boxes that are low level, maybe false positives. So we kind of ignore those, but when we see a lot in an area, we start to get suspicious, right? Could be another nematode, maybe it's SCN, maybe it's not, say inconclusive. These green ones are low level positives. These blue, so they're getting fairly high. These yellow ones, the old recommendations from most states was at this level, 10,000 eggs, you probably want to get out of soybeans for a while because your best resistant varieties are taking yield hits. And then we overlaid the red, so this is 20,000 plus, and these arrows that I was going to animate, they're pointing to some positives that are pushing west, right? There's virtually no sampling here. There's no negatives or positives, but we do know that there's some that are pushing west. And so it might be time to think about sampling in the future. And again, if you want to learn more about SCN, you can go to this website. There's a lot of good information and videos and things there.